warmest welcome to all of you here at the 4EU Plus Summer Symposium of Applied Embryology. It's a great pleasure to have all of you with us here today, and not just for the workshops, but also for the lecture day. I won't prolong this much, but I just have a few technicalities. Please, there are some snacks. Feel free to eat those whenever you feel like it. We're going to spend quite some time in here together so that you're not hungry. And then another thing, please, just for the sake of the organization, I kindly ask everyone who's present just to sign into the present sheet. That's from my side, and I'm very pleased and honored to have Mrs. Barbora Klusáčková here, who's going to start with our first presentation, and she's going to give a talk about the journey of a sperm cell. So thank you so much for coming.
Thank you, Kate, for the introduction. So I would like to share today something with you about my ongoing research about capacitation, but also I will tell you something more about sperm maturation. Today's talk is called Mission Fertilization. It might seem that the sperms are just a little tadpoles, but the reality is much more interesting. The whole journey of fertilization, or what it takes for the sperm to be able to fertilize the oocytes, is like an action movie. It's very, very interesting and the most important journey in the biology, I would say, because it's full of obstacles, superpowers, and it ends with grand finale, which is fertilization. So, what it takes for the sperms to be able to fertilize, it's not that easy, and we will talk about it today. So, we have to start with something that's like the origin story of the sperm, and that's spermiogenesis or spermatogenesis. Spermatogenesis can be separated into two parts, which is spermatocytogenesis and spermiogenesis, or also spermiohistogenesis. Cytogenesis is the first part, and it takes part in the seminiferous tubules. And we can see here on this picture how it goes. So basically, it transfers stem-like cells into very differentiated, highly specialized cells, which are spermatozoa. It starts on the basal membrane in the seminiferous tubules, where the spermatogonia are laid, and there are two types of them. There is type A and type B. Type A spermatogonia are the stem cells, and type B spermatogonia are those that will go to the differentiation. To multiple rounds of mitosis, type B spermatogonia will give rise to a population of spermatocytes of first type, and those will undergo mitosis. That what we can see is here. So spermatocytes of the first grade will undergo meiosis, and it will cross the blood-testis barrier, which is made by Sertoli cells. Sertoli cells are laid in the seminiferous tubules, and they nurture and they protect the spermatogonia and also all the stages of spermatogenesis. So, preliptotene spermatocytes will go through the blood-testis barrier, and they will enter meiosis 1, which is reduction division. Prior to this, there are chromosomes duplicated, and the preliptotene spermatocytes are still diploid cells. But in the end, we will get haploid cells, which will have only 1/2 of the original genetic information, and that will happen during the reductional division in meiosis 1. It starts with the prophase 1, which is a very long phase because it has 5 substages, which are called leptotene, zygotene, pachytene, diplotene, and diakinesis. During these, chromosome condensation begins in leptotene. In zygotene, synapses and bivalent are formed, and crossing over and between non-sister chromatids happens in pachytene. In diplotene, homologous chromosomes begins to separate, and they connect still in the chiasmata, and they are fully condensed in diakinesis, and also the nuclear envelope breaks down, and we are done with the meiosis one. Then we enter, no, we are not, sorry. We still have to enter metaphase, when there we have the metaphase disc, and where the bivalents align, and then the homologous chromosomes have to segregate to spermatocytes during telophase. And then we enter meiosis 2 with equational division. It will divide the spermatocytes further into 4, and we will get from one original spermatocyte 4 spermatids. But spermatids are still not done. Spermatids are those round cells, and they have to undergo spermiohistogenesis, which we have on the picture here. So spermiohistogenesis is the second part of the important spermatogenesis, and it has multiple different parts of what have to happen, right? And some of you saw that we saw like the highly differentiated cells which have like very small head and long tail. But how we get there from the round spermatid? We have to get rid of a lot of cytoplasm. Some of you already saw in the morning that some of the cytoplasm might stay there and form protoplasmic droplet. But that's natural, and we will get rid of it after the spermiohistogenesis, even in the epididymis. But mostly we get rid of the cytoplasma. Then we have to somehow get from the round, quite big nucleus to a highly condensed, elongated nucleus that we see in a sperm. So what happens is that normally in the nucleus we have histones, right? Those are the proteins that help DNA to condense. And as in normal somatic cells, we have the histones. But here in sperms, we need to change those histones to protamines. Protamines will not get there straight away. We have like proteins that will get there after the histones, and then we get the protamines. And thanks to these protamines, we will get highly condensed nucleus and also elongated nucleus as well, which also the structure called manchet will help to get this elongated shape. Then we have to form acrosome, which is very interesting and important structure in sperm. And sperms without acrosome would not be fertile, because acrosome is very essential for sperm fertilization. So how it happens? We have Golgi apparatus here, and the acrosomal cap is formed from Golgi particles. So it starts forming on the head of the or future head on the spermatozoa, and it covers the nucleus, again thanks to the manchet and intramanchet transport of proteins. We will get there the important proteins that need to be in the acrosome. Acrosome will eventually have to like disrupt the zona pellucida, so we need to have here proteolytic enzymes such as acrosome or acrosine or hyaluronidase and stuff like this. And then the most visible thing that needs to be done is form the flagellum. So it starts from the distal centriole, and then their microtubules start to form axoneme and also like other structures around the flagellum. And it's very important to for the sperm, because without the flagellum, the sperm would not be motile, and it would not never get to the oocyte unless we have a little bit of help from the embryologist. In the flagellum, we have a midpiece which is covered in mitochondria, and this is very important for the motility of sperm, because as we know, mitochondria are the famous powerhouses of a cell. So we need to really have them here because they make a lot of energy for the movement. So these are in a midpiece, and then we have other important proteins in the flagellum. Recent studies have discovered that there are over 1,000 proteins just specific for the flagellum of the sperm, so it really highlights its complexity. So here on this picture, we have all the structures we were talking about, and here we can see all of them. And I would like to just say something about equatorial segment and equatorial subsegment, which are very important things in my opinion, because it's where the sperm binds to the egg, and we have to really take care of and look at this region, because I think there are very important proteins located, and it's very important to study this kind of equatorial segment and even equatorial subsegment. For example, in boars, we see equatorial subsegment, which is like a triangular region here, highly rich in tyrosine phosphorylation, which is very important during capacitation, and it's very important for the further fertilization. So here again, we have head, we have midpiece, then we have tail or flagellum and endpiece. And again, here in the head, we can see the important structure of acrosome. On the acrosome, we can see that there are multiple membranes, right? Because the acrosome is like vesicle, so there's like outer acrosomal membrane, then there's an inner acrosomal membrane, and then we get to plasma membrane. Oh, plasma membrane is, sorry, it's on the outside. So we have plasma membrane, outer acrosomal membrane, and inner acrosomal membrane. And these structures are very important. We will talk about them in the acrosome reaction, but just take into note that there are 3 types of membranes here. So here we get to epididymal maturation. So from the testes, the spermatozoa have to go to epididymis. If we would take the sperm from the testes, it would not be able to fertilize the oocyte by itself. It would need to be done by embryologists, for example, by ICSI, because the sperms would not have like all the proteins needed for the fertilization. They would not be motile and stuff, because the motility really develops during the epididymal maturation. As you might know, the epididymis have 3 regions. We have caput, corpus, and cauda of epididymis, and these 3 regions either look different and also have different functions. So in the caput, we can see multiple little like tubules that are highly like densely coiled there, and then in the corpus we can see like there's only one tubule, but it's so coiled that you will not see like it's one. It would look like there's more of them, but trust me, there's one. And in the cauda, there is one thicker tubule that really looks more like straw or something, and it's pretty easy to extract the sperms from here. And so what happens is the sperm gain the motility or the forward motility, and their surface proteins are remodeled. Either they're getting new ones to them, because in the epididymis we have like epididymal epithelium, and these epithelial cells produce what is called epididymosomes. And these epididymosomes are extracted vesicles from the epididymal epithelium, and those epididymosomes have some of the proteins that need to be incorporated into the plasma membrane of sperm, and those proteins essentially then act as a binding proteins during the fertilization. And also, for what is good to have a cauda of epididymis is the storage of sperm, because if there is like collection of the sperm or like the sperms mature, and so they are collected here in cauda until the ejaculation. So what are those proteins I was talking about? So they have multiple function. As I was already saying, they have the function of fertilization ability. They will act as receptors or like binding colleagues of the receptors on the zona pellucida or even at the oolemma. And also there's like immune protectors, because there are a lot of possibilities of getting like infection and stuff, so we need to protect those spermatozoa. And also there are proteins that help with the motility in either during the passage of epididymis or after the ejaculation and during capacitation. So for example, epin is an interesting protein. It has a dual role. It acts as an antimicrobial agent and also helps with motility, because after the ejaculation it binds seminogelin and it acts as a little bit because it modulates motility after the ejaculation. Then we have HE1 protein, which is specific for humans, and it binds cholesterol, and it helps the fine-tuning during the epididymal maturation of the cholesterol levels, because the sperm has very specific membrane composition where the cholesterol is very important during further stages, and we need to have really fluid. We have to be able to either bind or release the cholesterol. Then we have CRISP1 protein and other similar proteins that act as proteins that bind to the zona pellucida, as I was talking about before. And another antioxidant agent that prevents oxidative stress in spermatozoa is, for example, glutathione peroxidase 5, which is specific for epididymis. And then again, for the help with the antimicrobial agent, we have lactoferrins and beta-defensins, which we have also, for example, beta-defensins in seminal plasma, but they are also involved in here. And those proteins that we have here, again, involved in zona pellucida binding, we will talk about in the end of today's talk. And here we get to my favorite topic, which is post-translational modification. So let me ramble here a little bit, because this is what I'm amused by. So mostly about phosphorylation, because this is my topic of my dissertation, or overall post-translational modification, but I'm mostly specified on phosphorylation. And post-translational modifications are very important in sperm maturation, because sperm, it has some kind of transcription and translation even after decondensation, but it's very limited. It's mostly in the early stages, and afterwards, it's the protein composition mostly changes by the post-translational modification. And post-translational modification act in all kinds of cell, and it regulates protein activity, enzymes, and stuff. So it's very important. So phosphorylation occurs thanks to the enzymes called kinases. And kinases takes phosphate, and they put the phosphate to something else, right? And this phosphorylation can either activate or deactivate the protein. So phosphorylation occurs on the residues, and these residues are either serine, threonine, or tyrosine. And for us, the interesting during the post-translational modification in the sperm maturation are mostly the tyrosine phosphorylation, because these are connected to the capacitation status and stuff. So phosphorylation. So here, either in the epididymis, post-translation modifications such as phosphorylation happens, and it affects, for example, motility, and it kind of happens mostly in the region of equatorial subsegment, which is the triangular region in the head of the sperm. Then we have glycosylation, and glycosylation we can see here. It's a very schematic picture of it, but what we can see here is that the sperm has on its surface something which is called glycocalyx. And glycocalyx is full of those kind of sugar residues. So we have those carbohydrate structures either bind to the lipids in the membrane or even right on the proteins. And glycocalyx mostly stabilizes the conformation of the proteins, but also it makes recognizable structures on the sperm. So thanks to this, we can have species-specific binding in the fertilization. And also those carbohydrates then help the sperm to bind to the oviductal reservoir and stuff. So it's very important, and it's very highly specific. And in every mammalian species, it looks a little bit different, which is what can be then used for what is used for species-specific recognition. Then we have ubiquitination, which is known post-translational modification. It controls the quality of the sperm during the transit of epididymis, and it helps to degrade those spermatozoa that might be not properly mature, or they might be dying through the apoptosis and stuff. So ubiquitination, what it does is that it checks for the wrongly folded proteins on the sperm surface, or like something that's, for example, like if there's phosphatidylserine, it tells you probably this cell is going through apoptosis, so it gets ubiquitinated, and then those ubiquitinated products are degraded in the proteasome. And sulfation influences interactions with zona pellucida afterwards, so it's very important post-translational modification, even though less studied. Then we get to ejaculation, which is like the transport from the male to the female, and we get the sperms to the really not friendly environment of female tract. So because of the unfriendliness of the female reproductive tract, we have to protect the spermatozoa because they need to survive there to be able to fertilize the oocytes. And what does this protection is seminal plasma. So what ejaculate is its combination of spermatozoa and seminal plasma. And seminal plasma is product of accessory glands, which are ampulla or seminal vesicles, the prostate and stuff. And those accessory glands produce different kind of types of important nurturing things for the sperms. So for example, we get from the ampulla fructose and citric acid, which really helps with the metabolic activity because it's like source for the energy in the sperms. Seminal vesicles again produce like energy sources, but kind of different. And prostate produces free amino acids and anorganic salts, which act as a pH buffer, which is also needed because there's different pH in the female reproductive tract than it's in the male reproductive tract or in the seminal plasma, and we need to protect against like quick changes of the pH. And then we have also bulbourethral glands. So as I was talking, it's nurturing, it's protecting, and it supports sperm motility, because once we get from the cauda of epididymis to the female reproductive tract, the sperm needs to start swimming quite a bit, because in the cauda of epididymis it was more like waiting to be ejaculated. It was not moving a lot, but after the ejaculation it knows that, okay, now is the time for me to go straight forward and get to the oviductal reservoir and bind there. So also, what is in the seminal plasma are decapacitation factors and other interesting proteins, again proteins, but the proteins get there mainly from the seminal vesicle and prostate, and they're very important. For example, here we have seminogelin, which acts with the epin I was talking about before. Then we have lactoferrin, clastrin, beta-defensins, again for the protection against the antimicrobial agents. And then we have spermadhesins. Spermadhesins are also otherwise called decapacitation factors. And those two names beautifully describe what those protein does. because sperm adhesins defines them as something that binds or adhere to a sperm surface, right? And decapacitation factors tells you that these factors will protect the sperms against premature capacitation. Because some of you already heard from me in the morning that capacitation is a very important step, but once it happens, the sperm lifespan will shorten. And the sperms really need to wait there in the oviductal reservoir and wait for the ovulation, so the fertilization will really happen. Because if they would capacitate before the ovulation happens, they would not survive this. Because they can wait up to a few days in the oviductal reservoir if they are not capacitated, but after capacitation, they will die probably pretty quickly. So, for example, those decapacitation factors in boars are AVN, AQN1, AQN3, also other is DQH, and those proteins have also the carbohydrates in it, so it's like glycoproteins, and they are acting as a protection against the decapacitation, but also they will also help with fertilization after decapacitation. Here we get to oviductal reservoir, which I was already mentioning, but what it really is. Here you can see it beautifully in this picture, and it's really looking like this. Like here is epithelium in the isthmus of the oviduct, and it has those structures on the surface, which I forgot the name in the English now. So these long structures are really in the contact with the sperm. And the sperms had the glycocalyx, right, the carbohydrates. And those before mentioned carbohydrates bind to something which is called lectin-like molecules. And those lectin-like molecules are perfectly binding proteins, like they really get together with those carbohydrate structures. But also we have like different types. We have lectin-like molecules on the surface of the sperm, and we have also carbohydrates on the epithelial cells. So it really works in a perfect concert, and it binds, and as it binds, the sperm knows that they are now like waiting for the ovulation. So here they're resting, they're not moving a lot, and they are like bind here, and they are waiting for the trigger of capacitation. And here we get to my favorite capacitation. So what it really is. It's very complex, and it's not easy to say in one word. So capacitation is a process that is regulated highly specifically at multiple levels. But I will try to sum it up pretty quickly for you. So what happens is there are changes in phospholipid distribution. There's something that is called lipid scrambling, which means that the lipids will change its position in the plasmatic membrane. So it will form rafts. Those rafts are mainly on the acrosomal side of the head, and there are specified regions where are the binding proteins very like concentrated, and also we have like high number of cholesterol molecules there, because from these rafts the cholesterol efflux will happen afterwards. And also what happens to the lipids is methylation, and it will transfer the phosphatidylethanol to phosphatidylcholine, and also some of the phosphatidylserine, which is normally the sign of apoptosis, will also occur on the outside membrane, outside part of the plasmatic membrane, because the plasmatic membrane has two sides, and normally the phosphatidylserine is in the lower inside side, and it flips to the outer membrane. Decapacitation factors are removed. Those are the proteins that are concentrated in the rafts, and it's easier for them to be removed. We can see that sialylation, which is type of glycosylation, is visibly reduced during capacitation, and the sialic acid normally is in the glycoproteins, which are the decapacitation factors, and as the sialylation is like removed, we can say that the decapacitation factors are removed, which helps either like to get rid of the bind between the sperm surface and the oviductal reservoir, which these decapacitation factors are the ones binding. And also it further promotes the cascades that happen inside the sperm. Then we have also changes in antigens, because the proteins have to like relocalize to this kind of region. We saw it on the previous picture, not here, sorry, here, because this is what the ejaculated sperms look like, but the capacitated have the factors more like here in the upper side of the head. So this is the changes in antigens, and we have also post-translational modification in the proteins. Again, most important, tyrosine phosphorylation in the proteins. And the tyrosine phosphorylation is something that really leads to hyperactivation. I will talk about it in a minute. And also we have some proteins that are adsorbed to the plasma membrane from the oviductal fluid. And we also have changes into lectin binding activity, as I was talking about. Terminal sialic acid is removed. And we have changes in the plasma membrane fluidity, which is caused by cholesterol efflux. But what really happens in the head and what happens in the tail is a bit different. So here on this side, we have something which is really like happening in the head, and here we have something that is happening in the flagellum. Because the things that happen in the head are those changes that have something to do with like decapacitation events, like the tyrosine phosphorylation and the cholesterol efflux, and so on. But here in the tail, we have to really like provoke the changes that will lead to hyperactivation. So what triggers the capacitation? What are the factors that like tell the sperm now really it's the time for you to capacitate? It's mostly progesterone, but also other stuff in the medium or in the female reproductive tract. We can invoke capacitation in vitro in specialized media, and this is what's done for like mostly scientific purposes in a research, but we can imply those knowledges into like what is really happening inside the sperm. So, for example, bicarbonate is present either in a female reproductive tract or in the capacitation media, as well as some kind of albumin. Here in the capacitation media, we have bovine serum albumin, and also there are other ions, for example, calcium and magnesium, and so on. So what happens is that the BSA or other type of albumin will provoke the cholesterol efflux. The cholesterol efflux means that the cholesterol from the membrane will be removed and the membrane will become more fluid. This will further help with the transport of the bicarbonate through the channels, and the bicarbonate will again it's like a really multi-action thing, because it will further again enhance the cholesterol efflux in the end. But what happens is that the soluble adenylate cyclase is activated, and the soluble adenylate cyclase does that the ATP, adenosine triphosphate, is transformed into cyclic adenosine monophosphate. And this uses protein kinase A, which is main player either in the head or in the tail. And the protein kinase A is a kinase which phosphorylates, and the phosphorylation happens by the protein kinase A on serine and threonine residues. But through this serine threonine phosphorylation, it activates protein tyrosine kinases. And protein tyrosine kinases further phosphorylates on tyrosine residues on multiple proteins, and it leads to capacitation. While in the tail, we have something similar. This cascade here is again the same as in the head, but also here the calcium is a very important player. Through Caspar channels, calcium gets in the sperm, and it activates calmodulin-dependent kinases, which again further activate the protein tyrosine kinases, and also it enhances dynein activity modulation, which helps with the motility hyperactivation. And also there is difference in a pH. It's in both head and the flagellum, but in the flagellum it again helps to the transfer of the calcium through the Caspar channel, and it's helping the changes in the hyperactivation. So here we have a little video of what hyperactivation looks like. So this is what normal non-hyperactivated motility looks like. Some of you saw the CASA system and you saw that it's not really this kind of straight movement, it's more like this, but overall the motility is kind of like progressive in one straight line. However, in the hyperactivated motility, the sperm really like moves crazy in one place, and then it moves to one another, something like what we can see here. So here are normally swimming cells, while here we will see hyperactivated sperms. We can see that the tail movement really changed. It's more like moving like, how do you say it? The tail is moving more like it's higher beat cross frequency, and it's driven by the calcium influx. Okay, let's move on. Okay, and now we get to the acrosomal reaction. The acrosomal reaction is triggered by the zona pellucida glycoproteins. So it means to normally, like for the acrosome reaction to happen, we need the sperm to get to the oocyte, right? Once it gets to the oocyte, it binds to the zona pellucida glycoproteins. In mice, it's ZP3, but in pigs, it's ZP4. And once it gets to these glycoproteins, it starts the reaction. So we have something called tethering and docking of the membranes. So during capacitation, the tethering happens. So the outer acrosomal membrane and the plasma membrane are getting closer together. That's the tethering. And during acrosome reaction, the docking happens. So those two membranes come together, and there are two like complexes. It's called V snare and T snare. It probably maybe in molecular biology you heard about it. And those are the same complexes that are involved in other vesiculation. And what happens is that the membrane makes the vesicles, and it forms those pores, and the inside of the acrosome will get outside. And as it gets outside, I was telling you that there are proteolytic enzymes such as acrosine and also like hyaluronidase and other important stuff. It will get out and it will disrupt the zona pellucida, so the sperm can get through the zona pellucida effectively and bind to the oolemma afterwards. So here on this picture we can see what the acrosome might look like, because we can detect the acrosome. I don't know if you here on the Embryology Symposium have seen it, but we can label the acrosome thanks to the glycoproteins and, for example, lectin PNA, which binds to these glycoproteins. So if you have like undisturbed acrosome, you will see this nice little cap on the sperm head. When it starts reacting, so this type of reaction, we will see something like this. There are some residues of the acrosomal membrane, but we will not see the beautiful cap like we see here. And when the acrosome reacts and everything gets out, we will not see anything left here. So, and we get to the end of today's talk, and that's fertilization. Fertilization might be like separated again into like primary binding and secondary binding. The primary binding is the species-specific binding. So here again, the aforementioned glycoproteins are important. The sperm will not bind to the zona pellucida if it has some different types of sugar residues on its outside, as it's those like partners on the zona pellucida. And then we have secondary binding, which is happening after the acrosome reaction, and it's bind those proteins that are in the inner acrosomal membrane. It's stronger and it involves inner acrosomal membrane proteins. And I was telling you that we will get to them. So those are the proteins that got into the sperm in the epididymis. So we can see really it's important for the epididymal maturation to happen, because otherwise the secondary binding would not happen, or it would be at least impaired. We have here P25B, which got there in the epididymal maturation, and we have also some glycosyl transferases and some other important stuff. And the secondary binding enables the zona pellucida penetration and fusion with the oocyte. Most important molecules for the fusion are UNO and ISUMO1, and also a lot of like CD molecules, integrins and stuff. So it's very important and very highly specific team, and I would not like to talk about it because it's very, very like not in my focus. So I will keep this one for someone else in some other talk. So thank you all for your time.
Thank you very much, Barbora. I myself have some questions, but I leave the floor to the students, whether anyone has something you'd like to ask.
Sure. Yeah?
Just want to say thank you for giving us a comprehensive presentation about the maturation of sperm. And I was just wondering, so you mentioned there's like multi-factors for sperm maturation. And what I know is for the infertility test for the couples is that you will check about like sperm's mobility, shape, and also the numbers, and also the healthy condition of the oocytes. But is there any check for the, what is that, the, what is the name of the, sorry, like the sperm will like temporarily store in the
Oviductal reservoir?
Yeah. Do they also check that to make sure that it can be successfully capacitated?
Oh, I don't really know. I don't think so. It's like, I don't know if you can, really like in the IVF center, I think this what I was talking about, the capacitation and stuff, it's not really like what they are dealing with, because they are using the sperms mostly like through the ICSI, so they really don't need for them to be capacitated, right? And this is like what should really happen. So this is what will happen in the female, but usually it's like if you go to IVF and you have to do the IVF, you will not need the sperm to be in the oviductal reservoir. Yeah.
Because maybe there's also like the issues maybe also because of the health
Yeah.
effects, so that makes it infertilized.
Yeah, but if it's like mostly when it's like a women factor, I think you go through the like in vitro fertilization, either like the standard IVF or ICSI. So this will not really be a issue there. Yeah. Yeah.
So just a question of understanding. So capacitation is like maturation of the spermatozoid. And so what's decapitation? At what stage of
Decapacitation.
decapitation?
Decapacitation. It's like the decapacitation is what happens from the ejaculation to decapacitation, right? It's not really like active process. It's just like it's the decapacitation factors are on the sperm and they're like protecting the sperm from the premature capacitation. Because if the sperm prematurely capacitate, it will not be able to fertilize the oocyte because it has to happen, the capacitation has to happen in the right time, right? And the decapacitation factors are those, for example, that bind to the oviductal reservoir. So if you have the AWN or IQN proteins on the sperm head, those are decapacitation factors, and those are the proteins or glycoproteins that will bind to oviductal reservoir. And the oviductal reservoir protects the sperm from decapacitation. It holds them there, and it will not let them capacitate prematurely.
So capacitation is the stage of maturation where there is this transformation of protein
Yeah.
on the spermatozoid, on the surface.
Also, yeah, it's like capacitation. It's really like the final maturation step, which will be like make the sperm be able to fertilize the oocyte. If the capacitation doesn't happen, it really would not be able to fertilize the oocyte. But if you take like IVF again and you add some kind of factor, you can invoke the capacitation, like not really naturally, but it can happen. But it's, yeah, I don't know if I answered your question. Acrosomal reaction is the marker or indirect marker of capacitation because only those sperm that were capacitated would be able to naturally undergo acrosome reaction. But like for example, when it was shown that sometimes like the sperms help each other, and they can like when one there is not like one sperm will get to the oocyte. It's multiple of them, and sometimes the first one will do the acrosomal reaction, but it will die of exhaustion before it gets to the oocyte. But it already like exposed the acrosomal insides, so it already disturbed the zona pellucida. So the other sperm that comes from this similar side will have it easier to get through the zona pellucida because it's already damaged, right? But it also happens in, like if you have it in the media and you try to like do it in vitro, you can add something which is called calcium ionophore. And if you add it in and you, for example, add it a little bit more of it, so there's more calcium in the medium than it should be, all of them will acrosomally react, even though they were not capacitated. But also sometimes the acrosome reaction of other sperms will like promote the acrosome reaction in the others. It's really like not the best marker for capacitation, I would say. Yeah. Yeah.
Thank you very much for giving us a lecture.
Thank you.
I have maybe a stupid question, however, quite naive. If you are talking about the premature sperm capacitation, is there something like a premature acrosome reaction?
Yeah, this is exactly the premature acrosome reaction. Like, for example, it could happen due to like mechanical disruption or something. Like, for example, if we are working in vitro experimental conditions, what we saw, for example, was when we really like weren't careful with the boar semen and we were like bumping it or something like this, or we have like one technician who was transferring them in a little bit higher temperature conditions that it's supposed to be, they acrosomally reacted. But it was not acrosome reaction that would actually happen after capacitation, it was unnatural and I would say immature. I don't know if it's like in the literature described as a premature acrosome reaction, but it's not a physiological acrosome reaction.
Does it mean that the acrosome reaction is more sensitive to the proper timing than the capacitation?
Not like, not like proper timing, I would say. Or like, yeah, proper timing, because if the sperms undergo the acrosome reaction before it's bind to the zona pellucida, it will not be able to get through the zona pellucida and fertilize the oocyte. So the timing here is very important. And also, like, if you have the sperm that underwent capacitation pretty early on, it went the long travel to the oocyte. It bound to the zona pellucida, and it exposed those insides of acrosome reaction. But it was too late because it was already like exhausted. It lost all the energy sources it had, and it will not be able to like get to the oocyte. So the timing really, like, it's all the concert, right? So the right time for the capacitation, right time for the acrosome reaction. Yeah? For the whole menstrual cycle, I would not say so, because the progesterone is what really like happens, right? What really triggers them in vivo. And the progesterone is there from the ovulated oocyte. Yeah, but I think like for the sperm to be able to get this trigger, it really needs the presence of the oocyte. I think like maybe it. It might be too late. Yeah, it's afterwards. Like if the ovulation occurs, then the capacitation is started, and it could be too late. Like if the ejaculation happens too late, then even the capacitation can happen too late, because there are right place for them to meet, right? So if the egg is already like somewhere behind the oviductal reservoir, you're screwed. You don't have the fertilization. Yeah.
I was wondering, so during the process that the capacited sperm swing to the oocyte, is it just like oocyte just like passively waiting for the sperm, or actually oocyte also did something to kind of like attract the sperm?
Yeah, it has, but it's not really like what I'm specialized about, so I don't want to lie to you, but it has some kind of like, there are the granulosa cells, and they also help like produce all the important like factors, but the oocyte like goes through the oviduct, and it has those like progesterone, for example, and stuff. So, and there's oviductal fluid, and oviductal fluid is full of important stuff. So, yeah. Yeah.
Hi, I'm wondering about the sugars that are around sperms.
Yeah.
How important they are? If there are some immunoreaction against the sugars from the female body, and if there is a possibility that the sperm can move even without them?
Move without them, I think so. But they are very like, they are important, because like, for example, when we see there's, for example, not decylylation, there's impaired fertility. But they're very like sensitive, and what was the question? Yeah, about the immune reaction. The female tract can have immune reaction against sperms, and it's type of infertility when the female has the like immune reaction. So I'm not sure if it's about the sugars, but I would say so, because like if you take a look at it, the glycocalyx is the most like exposed thing of the sperm. It's like what's really like outside. So it would be easy for the antibodies to get there, but I'm not sure if it really this is what causes the immune reaction.
I do have a few questions too.
Yeah.
You capacitation and the decapacitation factors. I've seen a phrase, for example, slow capacitating bore. So is the process of capacitation of the timing actually encoded within the sperm cell, or is it triggered by the environment where the sperm cell is located, or is it the two factors combined?
I don't know if you, I never heard slow capacitating bore before, but I heard about slow or early capacitation changes and fast capacitation changes. So not sure if it's this thing, but like there are like the capacitation is a whole complex process, and some kind of things take time, and some of them are very quickly done. So I would say, for example, the cholesterol is the thing that really takes time because it's a continuous process. And so I don't know, but if you think about it, it might be encoded in a bore, because bores, specifically bores, are very like heterogenic population, and they have very different proteomes, and even like bores, not species, but breeds have very different proteomes in it. So there was like study done on, I think, Landras and Duroc, and they were like comparing those two types against each other, and they figured there's like whole bunch of proteins different in them, and I think the proteins are like the main players in the capacitation. So I would really think maybe it's encoded in the bores, but again, I don't know.
Thank you. And one more question, if I may. You mentioned the importance of the epidermal maturation, so, and the proteins that the sperm actually acquires during the maturation. So in the case where we would have infertility and there would be testicular sperm aspiration,
yeah.
Do we have a way to make the sperm able to actually get the proteins when it doesn't pass through physiological epidermal maturation?
You would have to have like some, for example, ex vivo explants or something, but in a case of human, how you would get those, right? And so in this case, if the case is that you really, from the patient, need to get the TESA or something like this, it's like only way you can like be able to fertilize the oocyte through ICSI. Yeah.
Yes. So we wouldn't even try to make
I think it would like even like if you would take those sperm and like make them go through this like ex vivo.
Organoid or something,
Yeah.
something similar.
Yeah, but I'm not sure if it would be worth it. Yeah. Maybe in the future. Yeah.
Any more questions? Thank you very much, Barbara.
Thank you.
Well.
Thank you.